We've been talking about non-Mendelian inheritance patterns, right? Things that don't conform to Mendel's laws, but are still explainable by chromosomal theory. And also we've been talking about epigenetics, things where the genome actually gets modified and differences are actually inherited. So differences in phenotype, differences in characteristics are inherited, but there's not actually a nucleotide change. So we're going to keep with that theme and talk about basically the state of the DNA. How your DNA is packaged will affect what phenotype you are. If you think about being a heterozygote individual, you got a dominant allele and a recessive allele. If you're expressing both of those alleles, Mendelian genetics says you should show the dominant phenotype, because the dominant would be, would overpower the recessive allele. But you could be in the state where if you shut down the chromosome that carries the dominant allele, if you package that chromosome and condensed it way down so that you don't express that allele, you will show the recessive phenotype, even though you are heterozygous. This is what's called genomic imprinting. And shutting down one chromosome or another is actually inheritable. So if your mom shut down one of her chromosomes and that chromosome was inherited through the egg, then you might have an allele from your mom that she shut down, and so you don't express that allele. And so your phenotype is determined just by the allele that was inherited by your dad. You could do the reverse too. Males have imprinting, and sometimes the chromosomes you inherit from your dad might be in a shut down state. So you actually have the nucleotide sequence for the allele from your dad or your mom, but it's shut down so that you don't express it. This is called genomic imprinting. So maternal imprinting is where only the allele from the mother's chromosome is expressed. That would be the father's chromosome got shut down. You could also have the reverse, paternal imprint. Mostly it's maternal imprint that's most common, but there are instances of paternal imprinting as well. This is dynamic. So it basically only shows up in the first generation. So if your mom's chromosomes are the ones that are expressed and the dad's chromosomes are shut down, that is what all of your body is going to express. But when you go to make your germ cells, the imprinting is unraveled. Basically, you undo the imprinting, and then you imprint according to your sex, right? So you might not be expressing your father's allele, but then when you actually go to make your reproductive cells, eggs or sperm, you may choose to shut down one or the other. So you can actually see skipping of phenotypes, right? Something was expressed in your father's generation, you don't express it because you shut that chromosome down. But when you make eggs and sperm, you can unravel, and you might then allow that father's allele to be expressed in your children. So you can have a skipping of generation effect with this imprinting. So it's dynamic. It's undone in your germ cells. You reestablish it according to what sex you are. So if it's maternal, you're going to be shutting down father's chromosomes. Paternal, you'd be shutting down mother's chromosomes. This becomes significant in people who are in organisms that are genetically identical, right? This is some of the explanation why identical twins can look different from each other, because they're expressing perhaps different alleles, right? They've got the same genotype, but which genotype they choose to express might be different. Same thing with cloning individuals. You can actually have identical genomes. Do you look different from your identical twin? Yeah? Pretty significantly?
More as we get older.
Oh yeah, yeah. That'd be fun to like bring pictures and show you. So how do you actually imprint? How do you condense your genome in an inheritable manner? There's two ways to do this. One we've already talked about a little bit, but I'll review for you. One is called histone modification, right? The difference between whether a chromosome is compact or unraveled in a cell is a lot of it's due to what you've done to the histones in that little histone octamer that the DNA is wrapped around in the nucleosome. So the nucleosome is those 8 histone proteins with DNA wrapped around it slightly less than 2 times. And you can modify these tails. So one of the tails is really obvious in here. Here's one of those N-terminal tails of one of those histones. So there's 8 histones, there's 8 tails that are sticking out. And you can modify that by sticking residues onto it. They're stuck on at very particular sites. They're usually modified at lysine. The abbreviation for lysine is K, or arginine side chains. So any lysine or arginine that is existing in that amino acid sequence is possible to be modified. And you can modify it in a couple of different ways. You can do it by putting acetyl groups on it. This is acetylation. So there's enzymes called acetylases, and they come in and they can add or remove acetyl groups from the lysine or arginine side groups. You could methylate, you can put methyl groups on that. So methyltransferases are the enzymes that do this. They stick methyl groups or remove methyl groups. You can phosphorylate, so there's kinases and phosphatases. So you can stick phosphate groups on there. And you can actually stick several methyl groups or several phosphate groups. So you could single methylate a site, or you could double methylate or triple methylate. Same with phosphorylation. You could single phosphorylate. And then there's also proteins called ubiquitin proteins. And they're enzymes that stick these small little ubiquitin peptides on these sites as well. So you can acetylate, you can mono, di, or trimethylate, you could mono, di, or triphosphorylate, and you could also ubiquinate at multiple different spots on every single one of those 8 histones. So this gets complicated really quickly, right? 8 possible histones, multiple sites on each of the histones, multiple things you could do at each one of those sites, right? This is what we're trying to figure out what the actual code is for whether a chromosome should be condensed or not. And we're just trying to still crack what this actually is. We don't know what the actual code is. Certain things that get phosphorylated tend to open up. Certain things that get triple methylated tend to compact down. But we don't have real strict rules for that. So this is what's called the histone code. Did I put that on there? The histone code is trying to decide what are the modifications that would actually shut down a chromosome region and what would actually open it up. So just for nomenclature, you might see things like this. This says histone 3, lysine, residue number 9 is a lysine, so K9 is triple methylated. Histone 3, lysine number 14, acetylated once. Histone 4, lysine 20, methylated once. And so you could actually have a big, long list of what's going on at that chromosomal region. Now, this is just for one nucleosome that only has, slightly less than 2 loops of DNA around it. If you think about the entire chromosome or the entire genome, you've got hundreds of thousands of histone octamers, and they can all be slightly modified at all these different sites. This is really complicated and hard stuff to do, to try and figure this out. And the fact is, this is dynamic as well. Every single one of your cells can modify this and change it during whatever environmental conditions it's experiencing, right? So neurons have got certain histone codes to shut down regions of their genome, but other cells have shut down different regions. And the same cell in different time frames might be changing it as well. So getting into this real-time cell biology of what's going on here is really a difficult thing to do, but I want you to just understand that this is what's going on. Questions about this? All right. So what your mother or father has done to their chromosomes can be inherited, and so you might actually inherit some of this this histone code that keeps certain parts of your genome silenced. The second way you can imprint genomes is by directly modifying the DNA. So In histone modification, the DNA is not changed. You're just changing the proteins. In DNA methylation, you actually add a methyl group directly to the DNA, and it's added on the cytosine bases. So here is your normal cytosine, and then you change that hydrogen and you put a methyl group on it. So you're actually changing the base pair to be a methylated site. This is almost always inhibitory. If you methylate parts of the genome, that almost always codes for condense and package that DNA down really tightly, and so no genes will get expressed off of a methylated region. So there are DNA methyltransferases. So there's histone methyltransferases, those put the methyl groups on the histones, or there's DNA methyltransferases, and you could put methyl groups on the DNA. This can also be inherited. You can inherit modifications to the base pairs. And these methyltransferases will actually copy the methylation pattern when the cell is replicating. So one cell might have methylated its genome in a certain way. When you pull the two strands apart, a G was base pairing with that C, right? And it's only the Cs that were methylating, the cytosines. So when you replicate a new strand, that new strand may or may not get a methyl group, right? So the cell has to decide whether it's going to do that. So when you're copying your genome, not only do you have to copy the reverse complement, but you have to then remember, well, what was the methylation site of that C if I'm putting a new C on the complementary strand? So these methyltransferases, and there's a whole methylation complex, I guess, that comes in, and it reads the one strand and will put methyl groups on the other strand to make sure that you copy your genome properly and also copy the methylation state of your genome properly. So this can be inherited over time, right? This doesn't go away at every replication. But the new cell is free to modify it, right? So the cells, when a cell divides, it might have exactly copied its methylation state, but then one cell might decide that it needs to open up a certain region, and so we'll demethylate. So it is inheritable, but it doesn't always follow strict Mendelian rules, right? Because the cell could change it if it wants. Questions about DNA methylation? All right, the last DNA state is called X-inactivation. In mammals, if a cell has two X's, it almost always shuts down one of the X's and only expresses genes off of one of them. So this is X-inactivation. This is especially unique in females then, right? One of the X chromosomes, and it could either be the chromosome that it inherited from mom or it inherited from dad. Okay, we're not talking about genetic imprinting anymore. We're not talking about one of these chromosomes being inherited in a condensed and inactive state. We're talking about, okay, two functional normal X's get inherited, one from mom, one from dad, and the cell is going to decide early in development which X it's going to keep on and which one it's going to turn off. So this is X-inactivation. Most mammals do this. It's a random process in that either one of them could be shut down. But once you shut it down, every cell that is inherited from that original cell now inherits that state, okay? So it's like inheritance within your body, not inheritance from your parents. Does that make sense? What you get from your parents is generic. Early in embryonic development, the cells will decide, am I going to shut down moms or am I going to shut down dads? Once they do, every cell division that happens after that, the daughter cells inherit the same type of X inactivation. Okay. The inactivated guy, the inactivated chromosome, is called a bar body. And you can sometimes see it. Here's a little picture of a nuclei. And you've got some heterochromatin and some euchromatin. But here, this really condensed region of heterochromatin, that's actually the bar body. That's the condensed inactive chromosome. there's a lot of people still studying how one gets shut down and not the other, and how the cell decides to do it. What's involved are 7 known proteins and 5 RNA products. I just want you to know these names: XIST and TSIX, which is the palindrome, or I guess the reverse, not a palindrome, right? XIST, and then if you read it backwards, TSIX. These are non-coding RNAs that get expressed off of the X, and it's like a competition between the 2 X's as far as we can tell. One X chromosome will make a lot of these single-stranded RNA products, and they like go and stick to complementary sequence on the other chromosome, and so it's like a race. Which chromosome can make more of them? If the one chromosome makes more and early on, it will end up inactivating the other one. So if those versions get made on the one chromosome, they go shut down the other. And then there's kind of this dynamic process where they're both making them, and there's kind of a competition, and eventually one of them gets shut down. I don't know a ton about this, but I just want you to recognize it's non-coding RNA. It's RNA that doesn't turn into protein. And the RNA has complementary sequence, obviously, to the X, because it's an RNA that got made off of that genome sequence. And it will go stick to the other chromosome. So how that whole process works, I'm not sure, but it's like a little competition between them. What I want you to take home is that it's these 2 genes, and it could be either one. You know, the race gets rerun in every cell in early development, and so cells turn off. And once you do turn off, you're committed to that. It's permanent. Which makes for some interesting phenotypes. Mosaicism is where different cells in an individual's body express different phenotypes. Okay. If you're shutting off one chromosome and not the other in one cell, and the reverse is true in the different cell, if you're a heterozygote, you could have 2 phenotypes showing up in different parts of your body. A good example of this are calico cats. A cat's pigment is determined by a couple of things. First of all, we should say all the white stuff on his belly and on his paws, that's a different genetic mechanism. That's whether he makes pigment or not. So in a calico cat, the cat's body, the underside is usually going to be white, and that's not what we're talking about. We're talking about the tortoise shell, or this mosaic pattern that you see on the back, the pigment on the back. A cat can either make orange pigment, and that is from a dominant allele of the orange gene. Basically, the process of making melanin, you can either make it into a dark black product, or you can turn on enzymes and make it into an orange product called pheomelanin. So pheomelanin is orange, eumelanin is black. This is a gene that's coded for on the X chromosome. So if you have a heterozygous individual, and I'm sorry, this is not very good nomenclature. That's a big O, and that's a small o. So the big O is the dominant, it would make orange. A little o is black. So Mendelian laws would say every female that's a heterozygote is always going to be orange on her back. But since X-inactivation is true, that female, some of her cells are going to shut down the dominant chromosome, and so she is going to express just the recessive, and some of the cells will express the black pigment-making enzyme. Others are going to shut down the black and only express the O, or the big O, and are going to show orange phenotype. Now, it's not immediately, it's not the one cell fertilized zygote that decides what chromosome to shut down. It's actually a little bit later in development. So you've got a bunch of cells in this embryo, and it's at that point that all the cells independently decide which one to shut down. So the calico pattern, all of this black splotch here, that is because all of the cells are descendant from an original cell early in development that decided to shut down the big O chromosome. So all of these cells here in this patch all came from one original cell early in development. This orange patch, all of those cells came from a single cell early in development that decided to shut down the other. Decided to shut down the recessive chromosome, or the chromosome with the recessive allele, and so they're only expressing the orange phenotype. This is mosaicism. Different cells are showing different phenotypes. So you can have a homozygous female, right? She's only going to have the little o. That's going to be a black cat with a white belly. You could have a dominant, homozygous dominant. That's going to be an orange cat with a white belly. Only the heterozygote is going to be a calico cat. And also only a female, right? Males don't have this problem. You don't have calico males. Only very rarely do you have calico males. But
So what color are
well, males are either orange or black. Now, we're not talking about subtle different blacks and grays and spotteds and mottleds and all that. I mean, there's lots of other things that can be infecting coat color. But if we're just talking about orange or black, a male is only inheriting either, one X, right? So whatever X he got, that determines. And males don't do X-inactivation, right? Because they've only got one X already. Questions about mosaicism? Yeah.
kind of off the side, but I was reading about Down syndrome, so they kind of have this problem with like the mother.
Down syndrome, right? Yeah. So trisomy 21. Yeah.
So the mother mosaicism seems to affect on that.
Well, so yeah, the Down syndrome isn't because you're inheriting certain recessive or dominant alleles, right? It's just the fact that you have too many of the chromosomes. So you can have moderate Down syndrome as kind of a spectrum of how severe the issue is, and maybe that's due to the fact that the cells can partially compensate by trying to shut down one of those 3 chromosomes. But if you ever have more than one chromosome, it's almost always lethal. There's only a couple of chromosomal conditions that you can actually survive. Most of the time, if you got too many, the cells don't know what to do, and they just shut down, and the embryo dies. But trisomy 21 is a pretty small chromosome, and so you can kind of tolerate having an extra 21. I think there's trisomy 22. I think there's some instances of trisomy 10, but I'm not exactly sure all of these ones. But almost always, having too many chromosomes is lethal. And so mammals, I mean, there's certain mammals that don't do X-inactivation, but if you're a type of mammal that's supposed to do inactivation and you don't, it usually has problems because you're expressing too many genes on the X. The cell recognizes that as bad, and you have issues, or you don't survive. So. Questions? All right. We're moving on to linkage. Linkage is also a non-Mendelian inheritance pattern. Mendel was fortunate to pick peas because peas don't have weird, well, at least the traits that he looked at in peas, don't have weird mosaicism, right? The flower is either yellow or it's purple. There's no like mottled kind of purple, kind of white, or tortoiseshell, calico pea plants. There were distinct phenotypes, and all the distinct phenotypes that he looked at were on different chromosomes. So they segregated independently. They didn't interact with each other. Linkage is where 2 genes sit close to each other on a chromosome. And so the 2 alleles that are on the same chromosome tend to get inherited together. Okay? So it's not always that one trait always goes with the other trait. And we're going to talk about some examples, but with Mendel, it would be something like if a round pea shape and a purple phenotype. It's not that purple and round always go together. It's that a certain form of color and a certain form of shape go together. I think it'll probably just be clearer if we do an example. But we're talking about two alleles sitting on the same chromosome going together. Okay? So these genes will not assort independently. They will be biased, and certain traits are going to go together more often. If you remember back in my lecture from the history of genetics, I talked about Thomas Hunt Morgan. He's the guy in the early 1900s at Columbia University. He really is the one that adopted fruit flies as the model organism for genetic studies. He had a postdoctoral fellow named Kurt Stern. And I'm going to tell you about an experiment that Stern did. I was trying to look it up. I don't know if he actually did the experiment when he was in Morgan's labs, or if he actually did it when he took a position back in Germany as a faculty member. But it was Stern and Morgan that came up with this idea, this experiment. Morgan had found that certain traits inherit and are linked together with sex. He saw that white eyes tended to be more prominent in male flies. So he saw sex linkage, right? And so he postulated that chromosome that's determining sex is also determining whether you have white or red eyes. And so this was just when people were starting to actually be able to identify chromosomes under the microscope. You know, our microscopy skills were getting better. And so he postulated that genes are located on the chromosomes, and if two genes are located close together on a chromosome, they will tend to go together. Because he was seeing all kinds of other examples of linkages. And what people were doing mostly in those days was just breeding lots of flies and just collecting all kinds of abnormal phenotypes, like red eyes and white eyes, and we're going to look at these purple eyes today, and defective wings, and funky patterns of their bristles. And so people were just collecting all of these, watching the inheritance patterns, and then just crossing them, taking black-bodied individuals and crossing them with red-eyed or white-eyed phenotypes, and just seeing how they inherited. One of the things that made this experiment really unique is that people had just traditionally been looking at phenotypes, right? White eyes, red eyes, crinkled wings, non-crinkled wings. But we started actually identifying chromosomal phenotypes. And so this experiment is possible to demonstrate that linkage actually takes place and that recombination actually takes place is because people identified some chromosomal abnormalities. So here is an example of chromosome abnormality. In flies, the Y chromosome is longer than the X. Okay, so here is a male fly. This male fly has carnation-colored eyes, it's kind of subtly purple-colored eyes. It's a male, so it has an X and a Y chromosome. And the Y chromosome is longer, so we're going to illustrate it in yellow and then show this little extra length on it there. Okay, this is a normal-sized X. This is a normal-sized Y. But what they found is certain instances where the X chromosome was actually shortened a little bit. So you could actually start to identify chromosomes under the microscope and actually measure their length. And so they found certain individuals had a shorter chromosome. So here's one of those short chromosomes. This is normally how long it should be. This is indicating that there's a little break right there. So I've got a short X chromosome. Also, very rarely, but sometimes happens, in meiosis, the X and the Y will actually recombine together. Now, the X and the Y don't share very much sequence similarity, so this is a really rare event. But sometimes they get lined up in meiosis and recombination happens. So they found a chromosome that had an extra little part of the Y stuck onto the end of the X. And they knew that this X chromosome was short on this side, and this X chromosome is extra long on this side. So I've got 4 different phenotypes, I guess, of the X chromosome structure. You can have, well, I guess 3 different. Well, no, 4 different. 4 different. But you can either have a normal long X. You can have a normal long X with the additional Y. You could have a short X, or you could have a short X with a little additional part of the Y. This is the first experiment. Well, this was this work that was done in corn were the first instances of showing the actual recombination. But this one, for illustration purposes, I think is easiest to see. So what they did, what Stern and Morgan did, was set up a really clever experiment. They took 2 traits. We're going to look at the color of the eye and the shape of the eye. And then we've got these 2 different chromosomal abnormalities, the short X or this extra long X. And they were able to pair up and show that the inheritance of the chromosome determined your phenotype, and that it was actually crossing over between the 2 chromosomes. Okay, so we'll walk through this. They had a female fly. She has red eyes. Red eyes is the dominant wild type phenotype. And in flies, when we're talking about how to name the alleles, you just have, again, you have to get used to this. Fly phenotypes and fly genes are named after the mutation, the mutant phenotype. Okay? So the gene that causes the purple, the carnation colored eyes, that gene is named carnation. Okay? It's named after the mutant phenotype. Okay? So I have the carnation gene. This carnation means it's the recessive, and so that's going to give you the purple phenotype. The carnation gene plus means it's the wild type allele. So carnation plus gives you red eyes. Carnation without the plus gives you carnation colored eyes. Right? The second phenotype is the bar, or the shape of the eye. So here, this female has little kind of a weird shaped eye. Instead of being a nice round eye, it's got, we call it bar shape. It's got a little kink in it, kind of a abnormally formed eye. The bar phenotype is named so it's named, the gene is named bar because it's named after the mutant phenotype. It's a dominant trait. Okay? So big B is bar. This allele is the dominant mutant. Right? So if you inherit this allele, you will be bar shaped. If you inherit this one, this is B plus. That plus means the wild type version, and the wild type version is recessive. Okay? So B plus should, if you want to think in easier terms, you could just make it a lowercase b so that you remember it's the recessive. So B plus would be lowercase. What they did is they took a bar-shaped, red-eyed female that had the chromosomal abnormalities. She was heterozygous for the chromosomal abnormalities. So this chromosome that she has is mutant for carnation, mutant for bar-shaped, and is the short chromosome. The other chromosome she has is carnation plus. That's the wild type, so that's dominant, means she's going to have red eyes. B plus, the wild type bar gene, but that one is recessive, so she's going to show the bar phenotype, so she's got a barred eye. And then you've got the extra part of the Y. A lot of breeding went into generating that female. That's probably the hardest part of this experiment, was doing all the breeding to get that to be the case on her chromosomes. But what this does is allows you to set up what's called a test cross. A test cross is always crossing, it's almost always crossing a female to a homozygous recessive male. Okay? The male, in this case, is homozygous recessive, a homozygous mutant. It's not always recessive, homozygous mutant. No, I said that wrong. Always homozygous recessive. Homozygous recessive, okay. He's got the carnation allele on his X chromosome. That's the recessive mutant. He has got the wild type B. So he had normal colored eyes, but that is the recessive allele, right? So it's always recessive. And then he's got a Y chromosome. This male is going to contribute very little to what the offspring in the next generation look like, right? This is like genotypically, genetically, this is like the most impotent individual you could have, right? Because he's homozygous recessive, right? It almost really doesn't matter what he gives because he's going to give the recessive. What matters about the phenotype is whether the mom's going to give dominant or whether the mom's going to give recessive, right? And whether it's a male or a female, that's the only thing he's getting to determine, right? He gets to inherit, contribute the Y. But if it's a female, he didn't contribute the Y, so he didn't give anything. And if it's a male, all he gave was the Y, and it's the mother that's determining what that male offspring's phenotype looks like, right? So a test cross is wonderful because basically the male doesn't have, almost has no genetic contribution to it. Everything is determined upon what got inherited from the female, right? So the male is homozygous recessive, carnation eyes, which is recessive, and a round eye shape, which is recessive, and he's got normal chromosomes, okay? That, I know that was a lot to get in your head, right? I spent like an hour trying to go through exactly how I should describe this so that it became clear. So I want to stop. Are there questions about this? Are you clear about what the genotypes are and what the phenotypes are and why? Tamara, yeah.
The bar eyes?
Yeah.
That's dominant or?
The bar eyes is a mutation, but it's a dominant mutation. Yes. So B without the plus means it's the mutant and it's dominant. Okay. So it's going to show the bar phenotype and it's going to be dominant. Okay. So what are the gametes? It doesn't really matter what the male is, right? The male is either going to give a Y chromosome or it's going to give the X chromosome, and there's no real chance of recombination there. Recombination is such a rare event that we just assume that any sperm that this guy makes, if it's got an X chromosome, it's carnation and carnation colored and round shape that he's going to give. Right? So what the difference is what the female gives. And the way this experiment is significant is because we've got a marker now for all the alleles that the female has. That carnation, recessive carnation phenotype is linked to a normal X chromosome, normal side of the X chromosome. The wild type version of carnation is linked to having that extra Y. So under the microscope, you can actually say you can look at the chromosome and you can say what allele is existing on that chromosome, okay? The normal long X is going to go with the wild type round allele, round eye allele. And the mutant bar, giving a bar shaped, is going to go with the short end of the X chromosome. So this is the beauty of the experiment, is you can actually map on the alleles that this female has with an actual chromosomal shape that you can see under the microscope. Okay, so she can make 4 different types of alleles. We already know this from meiosis, but Stern and Morgan were trying to prove that this is the case, right? So if no recombination happens, then this female makes an egg that has this X chromosome, so she's got a short X chromosome, carnation, and barred eyes. If recombination does occur between those 2 genes on the 2 chromosomes, then the carnation, the wild type red eye will go with the Y. So, well, I guess let's talk about the other parental. If there's no recombination, the other one just remains intact, right? So we're calling these two eggs she can make. She is giving in those eggs the parental phenotypes. She's just giving her chromosomal contribution the way her chromosomes actually exist, right? So we call those the parentals because the eggs have the exact same chromosomal content that the parent did. The recombinance is if there was an actual crossing over event during meiosis. And so what will happen is the short arm with the bar body will get recombined with the red eye and the part of the Y. So you make a short X with an additional Y. The other one is going to be the long part of the X crossing over going with the normal. So you could have normal chromosome there. Does that make sense? It's just swapping genetic information in between those 2 genes, right? So the chiasmata crossing over event would have happened in between those. So
like the B plus gene will always be with a regular length
a regular red eye,
Okay.
unless there's recombination that happens. Right?
Okay.
If there's recombination that happens, that B plus is going to recombine, and so B plus is now going to go with the mutant carnation. Right? That's this guy.
But it's still going to be, B plus is always going to be
With-
regular length of that?
With the regular length of that.
Okay.
Yeah. Now, unless recombination happened on the other side of it, but we're ignoring that for now.
Okay.
Okay. So what Stern and Morgan did is they created these background flies, and then they crossed them. This looks difficult. It's not. It's not too bad. Because this is the beauty of a test cross. The male, let's look at the males. Here's the male's contribution, that male parent, and we'll just look at his male offspring, okay? Male offspring got an X, or I'm sorry, got a Y from their father, so we can just completely ignore what the father's X chromosome looked like. And we can just look at what are the phenotypic ratios of the males in this cross, okay? Their phenotypes are going to be dictated by what happened on their mother's X chromosomes. So if this male inherited a Y chromosome from his dad and a normal, well, a parental version of the female's chromosome. Right? This is the shortened chromosome that she has. Carnation eyes, bar-bodied, or bar eyes. That's what the phenotype is going to be in this male. Okay, funky bar eyes, and the phenotype is worse in males, so it's a smaller eye. So it's a carnation, bar-bodied eye, right? That's the parental. Just inherited the same chromosome, no crossing over. Or another offspring could inherit the other chromosome without recombination. So it will have the long arm, a long X with that additional long arm of the Y, and he will be red-eyed because the mother is contributing the red eye, and will be normal round shape. Okay? So when they did this cross, those are the most populous flies that they saw. They looked at all the males, and 99% of them are showing this phenotype. But very rarely, they actually did see recombination. They saw the two alleles that should go together, right? Because from the female contribution, the carnation eyes and the bar body or bar eye shape should always go together, right? But what they did see is sometimes the carnation eye was going with the round eye shape. And they said, what does that chromosome actually look like? So they went under the scope, looked at the chromosome, and sure enough, the carnation eye that used to be on the short chromosome with the bar allele on that short side, that chromosome now actually looks long, indicating that those two chromosomes must have recombined, and I must have gotten the long arm from the other chromosome. And I've now reconstituted a normal long chromosome. So you actually have chromosomal evidence now, like visual evidence, that recombination actually happened. What used to be a short arm with that carnation now is a long arm with the round-eyed phenotype. You could also look at the reciprocal. Here's this guy. This is a male that inherited the parental chromosome from mom, red eyes and round. The other one now has barred eyes. And if you look at his chromosome, you can actually see that it was a recombination event. He got one half of the chromosome with the Y and the red-eyed, now going with the short arm of the X chromosome that has the bar phenotype, the bar allele. You may have to go and spend some time with this. I understand.
So what happened with the one that's triple and not bar? The male right there?
This guy.
You said that his chromosome.
Yeah, this is a chromosome that did not exist in the mother. Right. The mother's chromosomes is either a short one with carnation and bar eyes, or an extra long one with red eyes and round. Okay? See that? Just look at it. It's short chromosome here, normal X chromosome with an additional long piece. This chromosome that this guy has, it's the normal side here from that chromosome, but it's the long part of this chromosome. So the only way that that chromosome could exist is if the two chromosomes from the mother actually crossed over and swapped sections. Right. The other is true as well, this one. The mother does not have small x with the additional y. Right? She's got long X with the additional Y, or a small x. So the only way to make that chromosome is to have had the chromosomes cross over. So they were able to link the actual chromosome shape with a phenotype, red eye or carnation eyes. That's what allows them to know which ones to look at, which ones to look for the chromosomes at.
Biola University offers a variety of biblically centered degree programs, ranging from business to ministry to the arts and sciences. Visit biola.edu to find out how Biola could make a difference in your life.